Yes, so my name is Olmo van Akker. I currently work in Berlin at the Quest Center for Responsible Research. But like Daniel said, I'm presenting work for my past employer, which is Tilburg University. I did my PhD there with this title and with this cover. So it's actually hot off the presses, and I will be defending in 3 weeks. And the dissertation is about preregistration and whether it works in psychology. So preregistration is basically when you publish your hypotheses, your research design, and your analysis plan before you actually conduct the study. So before you collect the data, before you investigate the data. So what does it mean that it works? Yes or no? So there's some debate about this. I think these are the two most important conceptual papers about preregistration, and 2 of the 3 authors are also in this room. So we're a good company. But basically, first point, preregistration increases transparency. So normally you only see the end product, right? You see a scientific paper with all the results in there. But with preregistration, you also see the plan. So you see the plan and the end product. So this gives you some insight into what happens in between. So for example, if there's a lot of differences between the preregistration and the paper, it could be that some shaky or shady things have happened. So it could be that that there's a good explanation for this, and that the researchers say, hey, we did it, we deviated because of this reason and this reason. So in that sense, probably the validity of the tests are not compromised. But it could also be that they say nothing about this deviation. And then anything could have happened in this black box, right? In this procedure between planning and doing the study. So it could be that they went on a cherry-picking expedition, right? Making some choices, deliberately so, to get to a desired result, typically a p-value below 0.05. So if they don't say anything about this, this can compromise, as Dai would say, the severity of a test. So because because of preregistration, we have the possibility to assess this severity, and that can also reduce bias potentially. So if I know that maybe my shady business or questionable research practices can be found out because people are checking the preregistration and the paper, I might refrain from doing that altogether, right? So in that sense, preregistration might prevent errors and might prevent false positives in the literature. So it can reduce bias. So in a nutshell, this is the conceptual grounds for preregistration. So how did we operationalize this? We basically said, okay, preregistration is effective if it has 2 things. 1, it should be producible. So preregistration should be producible. So this is a parallel term to what you probably know is reproducible. So reproducible means that there's sufficient information in the paper for you to redo the study, right? All the details of the methodology are given, etc. So producibility is that there is sufficient information in the preregistration to produce the study. So it's kind of a parallel term, but more focused on preregistration. So that's important, but it's also important that there is consistency between preregistration and paper, like I alluded to earlier. So, and the crucial takeaway here is that they are both important. One without the other is basically meaningless. So if I have such a tiny preregistration, it's not expensive at all, but I'm doing everything in line with the preregistration, that's still bad because there's a lot of things that we don't know about what's happening, right? There's no plan, so we don't know what happened in the meanwhile. Also, if I have a big preregistration, everything is in there, but I totally do something different, that's also problematic because I could have gone on a cherry-picking expedition, right? So we really need both these things in order for preregistration to be effective in reducing bias. So these things have been studied in psychology in isolation. So this is a paper by my PhD supervisor and others, Marjan Bakker, and they looked at preregistration specificity. So how elaborate are preregistrations? So we labeled that as producibility to make the link between reproducibility a bit more salient. And they found that they were often subpar. And this is another study that looked at consistency in isolation. So this is by Aline Klaassen and others. And they looked at psychological science papers and also found a lot of undisclosed deviations. So these things have been studied in isolation, What we did was we looked at them together simultaneously, basically. And what we also did was we used a very large sample size, larger than these previous studies. So we looked at two things, two sources, basically. One is a preregistration challenge. So this is an initiative by the Center for Open Science. It's already some years ago. And they gave $1,000 to researchers who published a study that was pre-registered according to their template. Then there's also preregistration badges that you probably are familiar with. So journals can hand these out if one or more of the studies in a paper are pre-registered. So these are basically our source of published pre-registrations. And in the end, we get almost 500 studies from it. So for all these 484 studies, we looked at the producibility and the consistency to see whether pre-registration is effective, yes or no. So what in these studies did we assess? Well, we basically looked at the empirical cycle that you can see here in circular shape. And we looked at 6 things. So the hypotheses, which are the building blocks of science, if you will. We looked at the design of the studies. So this is the operationalizations of variables. We looked at how researchers collect data collection procedure, how they analyze data, which is the statistical model, and how they interpret data, which is the inference criteria. So you could say that these things that we looked at are the most essential parts of a study, at least according to the empirical cycle. So let's go through these. So first of all, we looked at the directionality of hypotheses. So did the direction change from pre-registration to paper? So on the left side, you see pre-registrations, and on the right side, you see papers. And yellow is directional. So that means that an hypothesis was something like A is larger than B, for example. Non-directional is the light green bit, is that A is unequal to B. And the null hypothesis would say A is equals B, right? So these are different types of hypotheses. And we checked whether these were maintained in the papers. So what we found was that this typically did happen. So if you had a directional hypothesis, you typically had one in the paper as well. So that was good. But one thing that was really bad is this big thing, which says missing. So a lot of hypotheses that we found in the pre-registration, we identified, okay, this pre-registration has 4 hypotheses. Sometimes we could not find those at all. Sometimes we found 1 or 2, but a lot of times some hypotheses just went missing. So it could be that they selectively reported other results, right? This cherry picking thing, that's an option. It could also be that their use of language was so ambiguous that we just couldn't match the hypotheses in the pre-registration to those in the paper. So 2 things could have happened there. Both are, I would say, problematic. So this was the main takeaway with regard to hypotheses. What we also looked at was whether if an hypothesis changed direction, whether that was related to statistical significance. Because you could argue, okay, they change directions because they want to get that result, right? That statistically significant results. We did not find that, but this could, of course, be because this is a sample of pre-registrations only. So also researchers that pre-register, and you could argue those are more disinterested than other researchers. So they are maybe less likely to p-hack their way to a statistically significant result. But this is speculation. Here is a pretty flagrant example of a direction change. I'm not sure if you can read it properly. I'll just read it out loud. So the pre-registration said, we do not anticipate that judge attachment anxiety or avoidance will be significantly associated with judge accuracy. So this is a clear no hypothesis. Then in the paper it said, we anticipate that judges lower in attachment security, that is higher in attachment avoidance anxiety, will show lower accuracy. So this is just something they basically switched between pre-registration and paper. Pretty curious. So that's the hypothesis part. So here in one slide, you can see the other categories. The first 3 are the variables. Then we have the data collection procedure, the statistical model, and the inference criteria. And now I'm going to explain what these colors mean. So this is producibility, and it could be either not producible so that basically there's no information at all given, or only really little information. That is the red color here. That means it's not producible at all. And then we have 2 degrees of producibility: fully producible, dark green, and partially producible, light green. And that means that light green means that there's partially that some elements of this, the approximation of this variable, for example, in the left-hand case, are producible, but some aren't. And dark green means that it's fully producible, so all elements that should be part of the operationalization of the variable are producible. So what can we conclude from this is that for the variables, it goes pretty well, I'd say. So typically they are described in somewhat detail so that you can actually redo the study or do the study. The data collection procedure is fine as well. So this could be 2 things. They could either say, hey, we're going to test X number of people, or they could say how we're going to test people. So basically, okay, we'll be at this supermarket from 9 to 5, and every hour we'll take the first person and give them a survey. So the sampling frame, if you will. These could be 2 things.
And the gray color.
Thank you. The gray color is not applicable. So sometimes we had studies that didn't have a measured variable because they were experiments, right, for example. So gray just means not applicable. So basically, filter those out of your system. Statistical model is kind of a mixed bag. So sometimes it was specified in a lot of detail, but also we found a lot of times that things were missing, like the contrasts were often missing, like details were often missing that are still important, like how they estimate standard errors, those kind of things. People are typically limit themselves to the variables and the model, and that's it, without giving more details. And then we have the inference criteria. So here you see a big red blob, and that basically indicates that typically in preregistrations, researchers do not explicate what their inference criteria are. So they don't say, okay, we're going to use the threshold value of p is 0.05. So they just don't do that. They maybe implicitly that's the case, but we looked at whether they explicated this in the preregistration, and they did not. So the conclusions based on this. So variables and data collection procedure typically producible, a lot of information about it. Statistical model often lacks details, and the inference criteria are really rarely specified in preregistrations. So a bit of a mixed bag. So that was one part. Now on to the consistency part. Same categories there. Each bar is in the same category. And the colors are somewhat different now. So this time we dichotomized it. So either it's consistent or it's not consistent. So of course, green means consistent. Dark red means inconsistent. Gray again means not applicable. And now we have this nice salmon kind of color. And that indicates that we could not compare preregistration and paper. So for example, because it was not producibly described in the preregistration or not reproducibly described in the paper. So one of these fronts, information was lacking, which did not allow us to compare this at all. So we didn't find it fair to give them a 0 score. So we categorize them as uncomparable, basically. So again, variables are doing reasonably well, although you could say that every bit of dark red is problematic. But in the grand scope of things, they are doing relatively well. A lot of times they are just consistent between preregistration and paper. And then the data collection procedure, less so. And it has to be said that this is also because we were pretty strict. So if they said, okay, we're going to test 250 people, and they tested 248, it's still in this red thing. And the reason is that optional stopping can also occur even if there's small deviations from the sample size. So we want to be strict there. Better?
And these are deviations regardless of whether they're justifications for them.
Yes. Yeah. Yeah, that's not in here. Then the statistical model. Again, a mixed bag. So a lot of times it was consistent. A lot of times it was not consistent. I'll go into more detail about what was inconsistent later on. So this is more of the big picture. And for inference criteria, of course, there's a big salmon flavored part because oftentimes in the pre-registration they did not explicate it, so we could not compare whether they did the thing that they planned. Yes. So now closer look at the most common deviations. So this is the exact sample size that I talked to you about. So in 176 cases, they provide an exact sample size in the pre-registration. Blue means that it was consistent. And here's where the explanation comes in. So yellow means it was inconsistent, but they gave an explanation. Not saying it was a good or a bad explanation. We did not check that, but they gave an explanation. And red or pink says, okay, it's inconsistent, but they don't provide an explanation. So this is a problematic bit, yes.
Does that mean they don't disclose it, or they might disclose it and still be in the pink if they don't explain it?
Yeah, explanation is basically disclosing it.
All the pink ones, they didn't even disclose.
Yes, so we found a discrepancy, and they did not mention that discrepancy at all. So this is just mentioning it, not even providing a rationale, but just saying, hey, we did this instead of this. Yeah. So as I said, we were strict here because of optional stopping. Yes?
So what if they collected more data than they said in the previous
Same. It was also inconsistent.
So if they said something like at least, then it would be consistent? At least, and then a specific number of
I think we coded that as not producible, because if that's the case, you don't really know how much you're going to, or you should take into your study, right? It could be 200, it could be 250, it could be thousands. So it's not producibly stated in the pre-registration how many people you are studying. So that would be non-producible. There was a question there?
Yeah, I was just wondering what means exact.
Exact sample size basically means that they exactly specify how many people they're going to test in the pre-registration. So
is it like ±5, ±?
No, it's the exact number. So we are going to test 200 people. If they did 201, it's inconsistent. So this is the sampling frame. So in this case, they outline the procedure of sampling. And you see there it's better here. So a lot of times these sampling frames were consistent with what they planned. But again, you see this thing that will come back time and time again. They don't often acknowledge any discrepancies in the paper. Then a statistical model, which was also a lot of times inconsistent. So with exact model, I mean here, if they say in the pre-registration, okay, we're going to do a logistic regression, whether they actually did that, use the logistic regression in the paper, yes or no. So it's actually the choice of model, basically. That's maybe a better way of saying it. So you can see this was consistent a lot of times, luckily, because I would say it's one of the main things in a scientific paper or in a research cycle. And again, you see, if it was inconsistent, a lot of times they didn't specify that. They just used ANOVA instead of multi-level model and did not say why.
But this is not taken into account. Oh yeah, sorry. Yes. I was wondering about the
yeah. So this is the actual model, the choice of model, and we also looked at the variables in the model. So that's this part. Typically, they were aligned with the pre-registration. So the pre-registration said, okay, this is our IV, this is our DV, and this is our two control variables. Typically, those were in line with the paper. And here you see what the changes were. So an added control variable was often the case. A different IV was also often the case. And the other ones were less pronounced. So typically the added control variables. How many minutes does that mean?
1 minute.
In total?
Well, so we have time for
Right.
like some questions, right? So do you want to kind of...
Okay. So the consistency conclusions. hypotheses are often inconsistent between pre-registration and paper, as we saw. It is hard to compare pre-registrations and papers because the information is not always properly disclosed, either in pre-registration or in the paper. When information is available, typically they are consistent between pre-registration and paper, but they lack explanation. So they do not often disclose any deviations. So the final thing of my talk is, does pre-registration reduce bias, which is one of the end goals, I would say, of pre-registration. To that end, I want to first start with a study by Anne Scheel and also Daniel. Anne is also here, so we're in good company. So she looked at registered reports. And on the right side, you see registered reports. So those are basically pre-registrations, but the peer review takes place for the pre-registration. So before anybody sees any results, it's already peer reviewed. That's the whole concept of registered reports. So this is part of pre-registration, and these are standard reports. So just regular reports that are not pre-registered at all. So she compared those and found a way lower proportion of positive results for registered reports, which would make sense if you assume that a lot of results in the literature are false positives, right? So you could say that this is a more reliable picture of the state of things. So what do we expect from our study? So we also did this thing. We compared pre-registered studies with non-pre-registered studies. And we expected this, that our estimate of the portion of positive results will be somewhere in the middle. Because like registered reports, regular pre-registration prevents bias because people don't want to get found out and they don't engage in questionable research practices. But what registered reports have is also, it also prevents publication bias because the decision acceptance is made before the results are known, right? So we also have that preventative factor. So we would expect even less positive results for registered reports, medium for regular pre-registrations, and then the most for non-pre-registered studies. So that's our expectation. And we actually found exactly that for pre-registered studies. But our control group, we found also the same proportion of positive results. So red is pre-registered, green is non-pre-registered in our study. So basically, based on this study, there's no effect of pre-registration in preventing false positives. So I think that this has to do with the control group that we used. So what we did was we looked at pre-registered studies, and then we checked the related records, and we took the first non-pre-registered study to get more or less a comparable paper. And then we looked at all statistical results, except for checks of manipulations and statistical assumptions, because they are obviously not hypotheses. And then we checked, okay, how many of those are positive. So other papers did a higher proportion. They looked at all the first hypotheses as opposed to all hypotheses, or the most important hypotheses as opposed to all hypotheses. So you could argue those are p-hedged more, and that's why there was a higher proportion of positive results in that control group as opposed to my control group. So that is one explanation for this finding, but maybe we can discuss later. So finally, pre-registration does or does not seem to have the expected effects on the proportion of positive results. We thought it did, but based on this study, maybe we're not so sure anymore. And registered reports might be a better way to reduce bias. So overall, final slide, researchers need to describe their studies better in both pre-registrations and papers. Also important, researchers should explain whenever they deviate from the pre-registration to the paper. And finally, the evidence whether pre-registration is effective in reducing bias is not yet crystal clear, and more research is needed. Thank you.